delighted to introduce, and quite honored to introduce, Felice Frankel, who's a well-known photographer who's created many visually arresting images that help to convey complex scientific ideas and information to other scientists, as well as to the general public. Felice is a Brooklyn native who attended Brooklyn College, where she majored in biology. It turns out after she graduated, she worked in a cancer biology lab at Columbia University. Well, she started her professional career as an architectural photographer with, as I learned this morning, an old Nikon camera that she referred to as very good equipment that helped a very good photographer, at least in my view. Her lead fellowship period at Harvard in the early 90s marked her turn to science photography and many fruitful collaborations at Harvard, MIT, and well beyond. Many of us have probably seen her work on the covers of and in articles in journals such as Nature and Science, amongst others, without realizing it. Some in the audience may have encountered her 2006 book with George Whiteside titled No Small Matter, Science on the Nanoscale. Felice is currently a research scientist at MIT Chemical Engineering. She's been recognized through a number of awards, including induction as a fellow of the American Association for the Advancement of Science, a Guggenheim Fellowship, and the Progress Medal of the Photographic Society of America, which is their highest award. Today she'll present a talk titled More Than Pretty Pictures. Felice.
Great, thank you. What a crowd, man. I didn't know. Let me just say one thing, and then we could do that. Should I just press overhead when it went? Okay. Just that I'm thrilled. I had no idea if anyone would show up. I'm quite serious. You never know. Just by a show of hands, how many of you are not scientists? You don't matter. You matter more than anybody, really. Good. So I'm sort of talking to the choir in a way. So my whole thing is this. Excuse me. I want to make a case to you that what I do and what you do in terms of your representations is making more than pretty pictures. It's about your thinking, and I'd like to think I'm going to make that case later too, that you thinking about how to represent your work is a means of clarifying your work for yourself. But also, my whole thing, in a way, is about speaking to the public. We have to. We have to get them on our side, especially at this time of our country. We have to get them to pay attention to what we do and get them to be as excited as we are about our work. I think one way of doing that is through pictures. Now you can see the stuff. Okay. And please don't fall asleep, or try not to. Is this what we had before?
We can go one more.
Let's go one more. Do one more click?
Yeah, I think there's...
Yeah, a little, I think, these are pictures. You have to see the pictures, not me, right? Let's try one more.
Let's do video.
Okay. Okay. This is a picture that Alice Nesta took of her work. They are fabricating sort of bio-inspired engineering. They made this material that they were going to study connected with insulation of this kind of sea otter fur. This is a sort of fabric-like material, and this is Alice's picture. This is the picture that I made. Please tell me you think it's better, otherwise I'm in deep trouble. But it is the identical material. I just folded it and I lit it. That's it. I use my 105 macro lens, which I use pretty much most of the time. How many of you take pictures with a camera? Oh, good. So you sort of know what I mean by depth of field. If you don't, buy my book. So I couldn't get everything in focus, so I took a series of images at different focal planes and I stitched it together. It's an old technique. It's how you use confocal microscopy. It's how I got everything sharp. But basically what I did was I made a picture that I'd like to think is more compelling to look at and more important to get you all to ask, what in the world am I looking at? That's what I'm trying to do and trying to encourage you to consider doing. Here's a picture I took off of the web. I apologize for not giving credit. I don't remember who did this picture. It's a material called ferrofluid. And you know that ferrofluid is an oil suspension of iron particles. And so when you dress a magnetic field, which is happening here with a magnet, it does some interesting things. This is the picture that someone took. And this is the picture that I made of the identical ferrofluid. So basically, I dropped about 2 to 3 centimeter drop of the material on a glass slide. Under the glass slide is a yellow Post-it, very high tech. And under the yellow Post-it are 7 circular magnets. And you're seeing the ferrofluid respond to the magnets. If you look carefully, you could see the window pane from where the light was coming from. It was highly reflective, so I just decided to hold a green card above it. So that's where the green is coming from. And if you look carefully, you could see an example of a very bad photographer because I'm in the picture. Do you see the lenses or the double lenses? I didn't realize that that was there until it was printed in a book. George Whitesides and I did a book called no, that's the other book, On the Surface of Things. And the idea here behind this image is the following. We wanted to talk about magnetism. So why not start with an image that gets you to look? And now none of my pictures work without text. I'm not an artist. It's very important for you to know that I do not see myself as an artist. I don't know I'm a science visual journalist. I have no idea what to call myself today, but it changes with every day. But basically this is about getting people to look and to pay attention and to not be intimidated by science, and to then get them to be interested enough to ask questions. And then George explained it in the text. Luckily, I was very delighted to have that collaboration. It all started, this whole life of mine of 25 years of doing this kind of work, started when I was given this mid-career fellowship that you heard about. At Harvard, and I missed science terribly, and I sat in on a number of classes, audited E.O. Wilson, you're too young, but some of you might know who that is, and Jay Gould, and somebody suggested that I sit in on this other guy's class. He was talking about molecular biology. He was clearly very visual. And I went up to him after class and I said, I'd like to take a look at what's going on in your lab. Is that okay? He said, yeah, sure, yeah. Let's get rid of this woman, sure. So I go to his lab. They just got a paper accepted to Science. I looked at their pictures and I said, I think we could do a little better. This is their picture. You're looking at a self-assembled monolayer that has etched out hydrophobic lines in various shapes, making various shapes. So when they drop the water, it wet the surface at the hydrophilic areas and stopped at the hydrophobic lines. That was the science. That middle rectangle is about 4 millimeters across. That's the picture that I made, that they made, God forbid, yeah. This is how it was, shh, don't tell him I said this. And this is the picture that they made that was used in the article. And I said, as we were preparing, let me give it a shot. First of all, can you give me a little more interesting shape? Can you give me a grid, 3/3? And by the way, I'll get it in focus. And so not only did I ask them for that, I also said, what if we colored the water before we placed it on the SAM? And so this was the beginning of my thinking. In fact, if you think about it, I designed it so that the logo science could go up there. I was thinking I was looking for a cover, the audacity. Now I didn't like the etching. I felt that it was, you know, a little not necessary. And so watch the screen as I just turn the camera ever so slightly over and it becomes a whole different picture. And I thought, we've got a cover submission. Well, something very important happened. So just to remind you, this is a 3x3 grid. Those squares are about 3-4 millimeters across. I put fluorescing dye on the surface. They only had blue and green, not my favorite combination, but that's all I could do. And the editors said, it's not going to work because it's misinformational. We're seeing 2 colors of green and 2 tones of blue, and we don't understand that. Is that part of the science? That actually was really important that it happened, because from that 25 years ago, I realized that that's what I have to pay attention to keep the integrity of the science in every picture that I made. It was actually quite lucky. So I went back and got a better, tighter shot. And to remind you, those are 4 millimeters across. I dropped the water on the areas, the hydrophilic areas. The water stopped, and I suggest to you that this picture is maybe perhaps more compelling to look at, but it's more communicative. Their picture of their water drops, the caption said, "The hydrophobic lines stop the blending together." I'm showing you that. I'm communicating it to you visually. I'm showing you the chemistry. And yes, I'm showing off a little bit, and well, this is comparing one to another, and then this is what happened with it. So George said, "Stay with this, Felice. No one's doing this. You've got a future in this stuff." And he was right, with a little help from him, by the way, because Whitesides is somebody that people pay attention to. You can have your own opinions about the rest of it, but he's incredibly helpful when you're doing something right. And it was the beginning of my career. And there was an opening at MIT's Edgerton Center, which is where I started. And MIT is a place where they make things available to you, and if you're doing something worthwhile. There are things happening right now which I'm unhappy about on campus, but it will pass, I hope. It's a wonderful place. It's made me a home, and I'm very, very privileged. I work with some of the best scientists around and engineers. And so that started my career. And so I am trying to suggest to you that making compelling, honest images, which we'll get to in a minute, brings attention to your work. And I'm not embarrassed to tell you that I serve you. I serve the science community. Good science, and that's not a trivial caveat. For example, when Bob Langer and his colleagues were creating these devices, look at this. I mean, this was a little crazy. I took a very simple picture, and NPR, Wired, Scientific, I'm not just showing off to you. I'm telling you that when a picture is compelling and informational, it can help bring attention. Washington Post, Time, it's nuts. I was delighted, but I'm trying to make a point that what I'm doing ultimately helps you, and it's not about me. And this is a group of pictures of covers that I've gotten that first of all, I love doing that, sitting down with you guys and trying to figure out how to create an image to explain things that you cannot photograph. I'm going to get to that later. I put these words up there because how do you measure the value of covers? I'm trying to make a case on my campus that there is value in creating a center for the visual expression of science and engineering. This is my last-ditch effort to try to say there should be people like me, and animators and graphic designers and infographics people to be on campus to help you, just as I think that you should have it as well. But how do I measure that? I've been told by scientists that it is translated into funding eventually, but I don't have the data on it. So if anybody has an idea, let me know. But getting covers is not only fun, but it gets journalists to look at your work and then report it later, and then maybe even show it off to your funding agencies. I am convinced as I sit down with you guys to talk about what research, what kind of things you want to represent, it clarifies your thinking. Coming up with an idea of how to express it, not just by taking pictures, but thinking of a metaphor, for example. So let's just look quickly at figures and talk about what is the purpose of your figure. This is what I do in our master classes. Are you showing evidence? Are you showing structure? Are you showing a process? Are you showing change? Are you making a comparison? You actually probably don't think about that when you start your figures, because you make your figures after you have written the text and trying to fit in your data. I suggest that your thinking about your visual representation should be at the very beginning of your work, to work at thinking about how to communicate this stuff. And by the way, if you've made a decision that it's all of the above, you are in deep trouble, because you're showing too much. And that is something that I know, without even seeing your work, if we sat down with your figures, I know that you're putting too much in them. You are assuming that I am seeing what you want me to see. And here's the problem. You've been working on this material for months, maybe even years. You know exactly what to look for, look at, in your figures. And you're assuming that I'm going to see what you're seeing. And you're wrong, because I am the first-time viewer. Even if I'm an expert in your field, I am not going to see what you want me to see initially. And so here's an example of helping you see. And yes, I'm getting a little political, but you know, that's where my head is at these days. All right, the picture on your left is the inaugural cake in the Obama administration, the first inauguration. On your right is Trump's cake. The point of my showing you this is for you to see that the second cake is a complete ripoff of the first cake. But now let me, which was no surprise, but yes. But let me help you see it, okay? So what I'm going to first do, because I see that the lighting is under different, you know, pictures were taken under different conditions, let me just go to black and white for starters. They're starting to look alike. I'm going to go even further. I'm going to delete the background of one of the images. I'd like to think that you now are getting the point of what it is that I want you to see. The data of this image is the cake, and I'm suggesting I am not manipulating the cake. I'm trying to help you see the point of this comparison. We could do that with all sorts of tools, like using color. Color is extraordinarily powerful in your figures. Are you using too much? I'm going to answer that. Yes, you are. I know that you are, and you're using ugly colors. But let's look at Don Eigler's Quantum Corral. How many of you have seen this image before? Good. Phew. Then I don't have to explain that much about it, although to be honest with you, I'm not going to be able to explain the quantum effects going on within the corral because I don't understand it. But I do understand that there are quantum effects going on within the corral. And when we looked at this, my co-author and I, Angela DePace and I, for our book called Visual Strategies, When I showed it around to my physics friends, they explained to me that yes, it was remarkable that they were able to deposit atoms on a substrate, but what was also remarkable were, in fact, the quantum effects within the corral. And we're suggesting in this rendition, this is giving equal importance to the atoms and the quantum corral. The way it was colored, and we all know this is a visualization, of course, because these are numbers translated. Some computer scientist made the default picture colors this way, but the previous way. But what I'm suggesting is that the color can be a powerful means of getting you to understand what it is that you're showing. And in my opinion, the color was in the way. And again, we could argue that, but I'm just trying to get you to think, and this is what we do in the workshops, that color is extraordinarily powerful. Here's another example. The researcher, again, we're looking at two visualizations. The researcher wants us to compare the outlined area at the top. These are two explosions. The outlined area at the top and the outlined area at the bottom. And we're suggesting if you want us to pay attention to those areas, get rid of the rest of the color. Bring our eye to what you want us to see. The use of color can be incredibly exciting. Are you using it intuitively? For example, out of Klaus's lab years ago, this is an illustration where this was about the colored area are gels. And the gels, after you address a conductivity within the areas, the gels change pH. However, they colored it this way. If we're only talking about the change of pH among the gel areas, why not use pH colors and stay with a more intuitive approach to your decision about color choices? These are small changes, but what it is getting you to change your thinking a little bit about the way you present your material. It's small, but enough that I think the reader will get it a little better. Are you using the default in your software? Why are you letting some computer scientist decide on how you're going to color your material? Now, here's where I'm going to get some objections, I'm sure, because I always do when I show this. But Caroline Ross is working with AFM. And how many of you work with AFM? Okay, so all of you are used to seeing this pukey rust color. And you may quote me. You all use the default. And basically, we're looking at a grayscale image that the software is coloring. I'm asking you why, and this is rhetorical, why are you letting somebody else decide on what coloring you should use? And it gets in the way of the rest of the figures. So I'm hoping that I'm pushing you to go back to grayscale so that the rest of your figure could be more cohesive and aesthetically a little more interesting. However, as Caroline pointed out, and she's absolutely right, when you see a figure like the first one, you immediately know it's an AFM because that's been the protocol. That's what you're used to. I'm not sure we have to perpetuate ugly, but this is something that I would like you to actually just reconsider when you start publishing your AFM. Are you probably ordering your pieces? We're talking about composition now. Again, I'm using this from many years ago when Whiteside's group submitted to an NSF proposal. They wanted to show every conceivable self-assemblage they could come up with, captions, legends, all on one piece of paper. Help me see. So what we did, Angela and I, is we used their pictures and we ordered them in terms of scale. And we made a legend. If you want to know what A is, then you go to the caption and you could read it. It's helping us take a look at the details without being bombarded with just too much information. Are you creating a hierarchy, which is what I believe we just did in the previous image? We're showing you how to look. The first-time viewer has got to be shown a hierarchy of some sort. This is in our book called Visual Strategies. I mean, I'm not crazy about these pictures, but you know that that central image is the starting point, because we're showing differences in scale. And it all starts with that central image. That is the thing that you look at, and the rest of the images are associated with that center image. Not everything has to move from left to right or up and down. Sometimes you could get a beginning. Getting back to whenever we hold workshops, we show the work, and when we look at the work, I ask the group, what is the first thing we see? And you will be shocked, maybe not, at how many different answers you get. Again, you cannot assume that we are seeing what you want us to see. Can you start layering ideas? So this is from a workshop. 2 different situations that are very, very similar. Why not layer it to make it a little easier to compare? You don't have to go back and forth. It's right there if your data is layerable. Just made that word up. Edit out your graphical distractions. For example, again, this is opinion. I have no data on this. Parentheses, they're stupid. They're just another thing for your eye to look at. And also, do you need all those ticks that your software spews out? Give us some space so that you can put some more things together and it's not crammed into your figure. So back to Caroline, here's some data that MATLAB is, I think it's MATLAB, and all I did was I got rid of the parentheses and I deleted some of the ticks, just to give it a little more space. I have a thing with slanted arrows. I think they're distracting. You might disagree with me, but I'm going to try to talk you out of them. Here's an image that we used in our book. The researcher was showing the comparison between an aging brain and an Alzheimer's brain with slanted lines trying to show labeling. We made it into this image. And what we're doing is also we're adding another layer. We're showing this, and then overlaid with this. But notice how we labeled it with straight horizontal or straight vertical lines. Here's coming out of a lab recently in Chemie, and this is their image, and I just made a slight alteration to their lines for labeling by straightening them out into vertical. It's neater. Let me show you side by side. You might say, well, I'm splitting hairs. Just look at them really carefully. It's a cleaner image on your right without the slants. But I'm seeing some people who are not in agreement, but that's okay. Get out of here. Edit out other graphical distractions like bad labeling. I don't know why people do this, but for some reason, you like to put your labeling in rectangular boxes. I don't get it. What you're doing is adding an additional thing to look at. I suggest going back to simple labeling without the additional thing. Clean up your graphics. Of course, I'm going through this very, very quickly, so I'm going to try to make the point to you through my book. Edit out content. Now we're going into things that they're not just graphics, but they're about what you're showing. Edit out redundancies. In this particular image, figure, they wanted us to pay attention to the buffer layer. That is the important piece in this engineering. And so, going back and forth, I suggested if you want us to pay attention to the buffer layer, then just color the buffer layer. And when we see red on one spot, we're going to look at the red on the other area. So we make that connection, and we delete the image that is redundant. And so, again, this is not a finished image, but there is redundancy going on in its content. I'm showing you this because I'd love you to start paying attention to the world outside of this room and start looking at other people's graphics and start thinking that maybe it's relevant to our graphics. Who is going to tell me what this designer decided? I'm on the red line at Kendall and I'm going outbound to Alewife. What did this graphic designer decide to do in this graphic? I'm going to let you.
The word square.
Say it again.
Left out the word square.
Also left out all the other stations. You don't need them.
You don't need them. Thank you. That's my point. That's, I think, the way to go. I'm not interested in what happened before. I'm interested in where I'm going. And we didn't need the stuff from before. Again, you can argue, but I actually think it's fun for you to start paying attention to how people are graphically representing ideas in our everyday life. Also, edit out unnecessary data. This is what I usually sit down with my students with. They come in and this is the figure that they want to publish. So I start saying, oh God, do you need this? Do you need that? Do you need it in the body of the submission? Can you put it in supplemental? And eventually, after going back and forth, this is what we wound up using. I made a new picture of their chip. Notice that I put the scale in the image and used it as a design tool, not just a bar. And I put the SEM in an illustration. And if you don't agree, that's fine, but I think that this second one is a better figure for the journal. And we'll wind up with this section in showing you a sort of process. Again, this is out of Klaus Jensen's lab. They were looking at a device which had charcoal in it, inside, that's the granular stuff. And it was a very long device, so I took a picture of one area, the front and the back, and they put the two together in this image with a fuchsia, if you'll excuse the expression, representation. You know, architects use this when you want to show a corridor that's longer than you have room for. And this is how they put the labeling and other material in it. Okay. The first thing I did was, please give us more room to breathe. So I got more space around the perimeter. I then am suggesting, and some of you are going to have a knee-jerk reaction to this one, I think that labels do not need caps. I think all of your labels should be lowercase. It's cleaner, unless it's something that needs to be uppercase, but it's cleaner. And also, getting back to the slanted arrows, get rid of them and get rid of the arrows per se. And here I am with this new rendition. In my opinion, cleaner, lowercase, no slants. And there's one more thing that I suggested they do, and who's going to take a hard look at this and tell me what's not quite right?
Red lines.
Yeah, I'm not crazy about the red lines either, but they insisted, so let's keep them in there. I agree.
You decreased the font from the previous picture.
Yeah, well, let's see, is it decreased? Yeah, well, okay.
That helps. That's better.
What is better? Which one?
This one.
Thank you, okay. But there is one other thing. Yes?
Is it the catalyst section lines?
No.
Okay.
Yes?
Does the blue color communicate anything? It doesn't, I guess.
It doesn't, but that is the real color. That was real. So I'll give you a hint. You ready for a hint? Mm-hmm. It has to do with the inserts.
Oh, orientation.
Very good. The orientation of the inserts. The inserts are SEMs. They're not matching the optical image. This is it. However, here's my other issue. Do you need the SEMs? Is it giving us any more information than the optical? No. But you want to show that you took SEMs. Right? No. That's the hardest thing. Take a step back. Do I really need this piece? Even though I worked 2 hours on getting these damn images, do I really need them? And I suggest that you don't. So, and that's a hard thing. I promise, I understand that. So this is the comparison. If you don't agree, get out of here. I would like to make this thinking collaborative. This is what we do on my campus. There is nothing better than finishing a graphic. I was asked this today at lunch. How should we do this? Finish your graphic and then show it to your colleague. A colleague who is not threatened by your work and who will be honest and who will be a first-time viewer. You have to show it to somebody who is not familiar with your work, or at the very least who has not seen what you've been doing. Because you've seen it too much and you cannot read it. It's like writing an article. You've just been too absorbed in it. You can't see it any longer. I do this myself. So this is what we do on campus. We sit down, we look at people's work, and as a group, we dissect it. Sometimes when we're really lucky, we bring design and science and engineering people together. We did this at Harvard, where we brought GSD people and scientists and talked together about looking at their work. I am told that this kind of representation that's readable and compelling helps with funding. Why not? If you have an executive summary to your program and there's a space for your graphics, an executive summary graphic, why not make it readable as opposed to putting everything you can in one graphic and nobody wants to bother reading it, and making it a little more comprehensible and more clear in terms of seeing what it is that your submission is about. If you're given real estate for graphics, don't leave it for the very end. Think about it just as much as you're thinking about the text. And finally, as far as this section is concerned, this is my thing. My thing is about making science accessible. Yes, and I'm being obnoxious with this filter, but making it acceptable to the public. We have to do this. I also think it helps you think about how to represent your stuff. So quickly, I'm going to go through pictures in my new book, which I hope you take a look at. The first chapter is on using a flatbed scanner to produce images of science and engineering. Yes. How many of you have used a flatbed scanner to capture a device or something of your work? A few. Good. It's crazy wonderful, and it's cheating, but it works. So Stefan came to me one day from Meki, and he was creating all this material that had different physical characteristics, and he wanted me to get a cover. And I said to myself, this is crazy. But what I did do, I started putting the pieces on my scanner. This is an image made with a scanner. I'm not using a photograph. And eventually we got a cover. This took 30 seconds. It was crazy, and that actually started my craziness. No, I was always crazy. So this is a 1 centimeter device that I plopped on my scanner. This is with microneedles. It's going to address perhaps vaccines. This is an image of Euplectella that I made on my scanner. Now I'm not making a new image here. I'm zooming in to this 1 gig file. You can see, yes, and it took a little bit of scanning, but not crazy. It was something like 10 minutes. But look at the detail. Look what nature does. It's quite remarkable. Those fibers are 50 microns. That's the kind of resolution you could get on a scanner. Not a very expensive scanner, But you have to have a scanner that you could set the DPI. If you can set the DPI, you can get this kind of resolution. You can put an egg on your scanner. You can put pears. You can put tomatoes on your scanner. And look at the detail. It's fabulous. I'm telling you, you're going to have a blast. But it really, but it's also a means of documenting material that you could publish. In the course that I gave a couple of years ago on edX, and you could find all of the work, all the videos on OCW, OpenCourseWare. One of my students created software that told the scanner to capture an image every 20 seconds. And he poured a supersaturated solution of sodium chloride, and this is the movie that he got. Not bad. That's what you could do with a scanner. The second chapter is on camera basics. I have to tell you, I am flabbergasted at the incredibly expensive equipment on my campus of cameras, ordinary SLRs, and they have no idea what depth of field means. It's fascinating. And so I'm teaching you the basics of photography, the differences between shooting at f4 and f32, for example. I talk about what I say take two. I compose easier if I have two of something. I don't know why. Maybe because I like to show that you could produce more than one of your devices, but it really does help in composing. I talk about backgrounds, how your backgrounds, if they're carefully chosen, can enhance the information or just make it more interesting. This is, is this bee subtilis growing in a petri dish? I took the petri dish, put it on a blue background, then I put both of them on an orange background. Here, I'm trying to show somewhat of a similarity in the way light responds, that is the way light responds to the material, by using a CD to show a similar phenomena, but unfortunately a lot of people don't know what a CD is anymore. So I mean, this is my problem getting, and sometimes I actually showing the sketches of creating a hand suggestion. I like showing the human aspect of this highly technical material by showing the sketches that the initial devices were considered. This is a background. I knew someday that I was going to use these watercolors as a background. I got it at one point. I was imagining how I was going to use it, and finally it happened. For the book No Small Matter, George wanted me to take a picture of a drop of water. And he was going to explain why exactly are we seeing a drop? What's the molecular configuration of that? And so for you photographers, I'm using a 105 lens, and I'm focusing on the drop. And so therefore, everything in the background is going to become out of focus. If you look carefully at the drop, you could see it's acting as a lens. That was unintentional, but you could see the watercolor palette focused, which was fun. And so it's about using backgrounds intelligently. Chapter 3 is about light. Photography is all about light. I mean, I could write books on that. And so I'm showing, we have an interactive device where you could actually play online and just see how putting a white card next to the light source is a fill, and all these things that I tried to explain. This image I took with a light table as the light source. It wasn't on a light table. The light table was the source of light, and here is the same device, and the source of light was the scanner light. Why not? That's the fun. You know, that luckily I'm still having fun. When I'm not having fun, I'm not going to do this anymore. But I'm still having fun. Just trying to show you light is what photography is all about. Here, I put a fishing reel, because this is, Pecco is doing work on sports. I put it in a garbage can, and I added light from above. And look at, such simple idea just by adding light from above, and it changes the entire image. By the way, this is the garbage pail that I, just in case you're dying to do this. Here's a detail of material out of one of the labs where wafers are deposited. And watch the screen, just as I add some light from above, it changes the entire picture. I mean, that's what you do in photography. You just don't take the first shot. You move things around. You move it around, you move the light around, you move the background until you get something that is very, very definitely better than the first time around. So I talk about direct lighting, different from diffuse lighting. Here, again, because I was still working with George, this is PDMS that had etched in it a grating, which when shown with light, it separated into the wavelengths, but you don't see it so well. So I put a shadow in the image so you could see it better. And none of this is written anywhere in a book. This is all about thinking. You could do this too. I'm not doing anything that you can't do. I really believe that. That's why I wrote the book. I want you to do this for your work. It's worth the while. And if you don't have the time, at least maybe I could start introducing standards to you so that when you hire somebody to do it, you know what to ask for. This image was something that I took many years ago, which other people use without my name, but I'm not going to touch that just yet. It's very annoying. These are quantum dots, and they're cadmium selenide nanocrystals, and out of Moonji's lab. These are vials that contain the same material. The only difference is the size of the nanocrystals change the fluorescence. That is the wavelengths in which they fluoresce. So that's the only difference, that the size is changing the wavelength under UV light. And I thought I was being very clever in making this image. Show me with your hands where the vials are and where the camera is by looking at this. Okay, and why do you think the camera, why do you think that?
Because of the air bubbles.
Thank you, because of the air bubbles. I thought I was being subtly clever with the air bubbles. They hated it. This is the picture that they asked me to make, which is very disturbing. But now they finally came around 15 years later, and they liked the other one better. But basically, again, you're seeing vials that contain cadmium selenide, but just different sizes. Then they're fluorescing at different wavelengths. And yes, I'm not showing off, only saying JPhysChem never puts photographs on their cover, but this is the data. And just keep in mind that the reason why this works is that all you see are the vials of the material. There's nothing else in this picture. You're not distracted. You're seeing the color changes. That's what this picture was about. In chapter 4 about phone cameras, I have to say the book is based on, the book is based on a course that we put together. It's usually the other way around. But this is based on a course. And in the course, when I discussed phone cameras, I say, you could take as many pictures as you want with your phone, but they can't be very serious. I'm just telling you right now. I've changed my mind. It's ridiculous what you could do now. Who wants to guess what you're seeing as this is the chapter opener of the phone camera section. Well, I'm a foodie, and I was cooking various colored peppers, and I put the glass cover on top, and I got this within 20 seconds, and then it was gone. But you're seeing condensation and optics and all kinds of fun things. You can do amazing things. And it also has to depend upon what frame of mind you're in. I was walking on ComAv, and I just was talking the previous day about self-assembly. And so I'm seeing these drops self-assemble. And what you're seeing is a tree that is wrapped around with cellophane. I have no idea why they're doing this, but the bottom line is I saw these self-assembled drops, and I took a picture with my phone because I don't carry around a camera. You can quickly show your colleagues your equipment by putting white paper all over the place of your equipment and then deleting the seams. This literally took 2 minutes, and it's a visualizable apparatus. But keep in mind, still today, it's changing, but the file size of your phone does not compare to an SLR or your scanner. This is a petri dish with E. coli growing under 4 conditions, taken with my phone. This is the picture that I made with my scanner. When you zoom in, the phone is just not as resolved as the scanner or your phone or your camera. It's not there yet, but it will be. Probably they have it in the back room waiting to sell it, but it's very exciting what you can do with your phone. I've used this device to put my phone in my ocular in the microscope. It really does work quite well. It holds it exactly in the right position. Here I'm just playing around. These are coffee bubbles in a glass dish, taken with the phone with that device. Just really quick images. The 5th chapter is on microscopy. I am not a microscopist. There are people who know much more than I do. But I use it as a photographer. And so I take you through how showing various scales of images in terms of size with a microscope. Here, Jerry Fink allowed me to grow yeast colonies in one petri dish. They differ in the morphology and the genetic component of them, and that's how it translates into the morphology changes. But in my opinion, this is not a successful picture because you're paying too much attention to me, the light. I'm not an artist. I don't want you to pay attention to the photographer. The light is too much in the way. And so I think that this is a much more successful image for you to make that comparison. Here I'm using Nemarsky differential contrast that helps in emphasizing surface structure. I've included a few other people's pictures of confocal microscopy because I don't do that, but I wanted you to know that it's an instrument that's available. In the chapter 6, I talk about presenting your work. I talk about movement. Now watch this is the Belozov-Zhabotinsky reaction. I'm clicking off a series of still pictures. And now I'm showing you juxtaposed next to each other. Film, as you know, well, maybe you don't know film, but movies are single images stitched together, so that we're used to seeing moving pictures. But there is a value of seeing one moment next to another. The BZ reaction, anyone familiar with that? Okay, so you're seeing self-oscillating chemistry with malonic acid as an indicator to see what happens as it progresses. And here I'm showing movement in a still picture. And even though this is not in my book, but I just did this for the first time, I still am not into video, but I took a series of still pictures that we stitched together to make a movie. I'm going to get into video at some point, but the beauty of this is that you get a high resolution. Every image is 36 megs, so if you want to go into detail, you could zoom in and you could see things. I talk about time. This is an example that I think is not my picture. I think it's very interesting where these photographers went to the same spot and photographed the same area as Ansel Adams did. And so they collaged different moments of time in one picture. I don't know if you could translate it to your work, but I think it's wonderful to actually think about. And I talk about scale, and inserting, instead of a bar, inserting a nice clean scale line. And if you insist on using that dime, at least compose it in a more interesting way, please. I beg of you, right? Okay. And I touch on abstraction and metaphor, how thinking about how to create a metaphor to explain something that is not visualizable. For example, the way a CD and an SEM I made years ago works like a piano player roll or a music box. It goes on, it's talking about, the idea is to describe what binary is about, binary language that computers talk to each other with. It's on or off, 0 or 1. In my former life, as I mentioned, I was an architectural photographer, and I have to have 1 or 2 pictures. I have thousands, I've got to use them somewhere. But it basically is Faye Jones created this beautiful pavilion in Mississippi. You cannot tell where the landscape or the architecture end or beginning. In addition, he abstracted the pine coat surface. He didn't copy it. He abstracted the essence of the pine coat surface into his columns. I think it's absolutely beautiful work. This is an abstraction or metaphor that's wrong. And people actually for years have thought, and maybe still do, think that an atom looks like this. It's dead wrong. So you have to be very careful. Whereas Roger Hayward, his metaphor or abstraction of an atom is suggesting the probabilistic nature of the electrons. So it's very exciting, I think, to start coming up with these ideas when you start speaking to your public, but also your colleagues. I decided to put this in fast. I took out the text temporarily of this metaphor, where I'm now going to insert the text. This was at the New York Times. This was right after 9/11. I think it works. Almost done, I promise. I talk about image enhancement. This is not discussed enough. I'll tell you that when I started writing this chapter, I went to our Office of Scientific Integrity for their material. All they talk about is plagiarism. They do not touch on image enhancement. How far are we permitted to go in science, if at all? Always maintaining scientific integrity. This picture, a Hubble image, is completely fake. People think the universe looks like this. Now, I think it's perfectly fine to color images as long as we say that we are coloring our images for the communicative value of it. Take this example of mine. Jerry Fink had me photograph his gorgeous yeast colony. It's absolutely stunning. Watch the screen as I digitally deleted the Petri dish. I wanted you to see the amazing structure of this yeast colony. I suggest that I am not changing the data. The data here is the morphology of the yeast colony. But I indicate that I've done this. And he was happy. He didn't like it too much until we got the cover, so that was enough. But here's another example. This is for a book that George and I did. We wanted to show these amazing Proteus colonies, how they're growing in these gorgeous colonies. Watch carefully as I digitally delete the cracks in the agar. I'll go back. Was I permitted to do that? We can talk about it. I say that I've done that. I wouldn't have done that for the submission in the article. I would do it for the cover. And the editors agreed with me on this. But look at this. Oh, and I also colored SEMs, which is a lot of us do that too. But we indicate that we've done this. But here's one for you. These are gels that have absorbed fluorescing dyes. I took this image on film. FILM, right? You might have heard of it. It turns out that this film did not read the orange wavelength. This digitally fixed image is what my eye saw. The digitally fixed image, which I colored later, is more honest than the first image. Let's talk about that one of these days. But it's something to think about. By the way, the Nature editor, I had a conversation with Nature and they said there's no way they would have accepted that. So, but you know, I think it's something to think about. And finally, in the end, I show case studies where I show how I start with an image, and this image they liked, I stretched it, I took a syringe on the flatbed scanner, I inserted it into the picture, I took their fluorescing cells, inserted it into the drop, and we got the cover. This is a completely fabricated image, which I say, it's a photo illustration. I cannot draw for beans, but I can take pictures. And so what I do is I take pieces of pictures and put them together to represent things that you cannot actually photograph. Here I started with an awful picture I made of this plexiglass, sent it to the Nature director, told her what I had in mind. I put the graphene on top. She then came back. This was a cover about epitaxy, creating copies of material in the presence of a graphene layer. This was her idea, and this is what these are all pieces of pictures that I made of stuff, and we got a lovely cover. At the end, I have a visual index, whereas that's where I go into the science. Because I don't want to distract. This is a book of how to make pictures. And I wanted all the science to be in the back of the book. All of this stuff is on OCW. Honestly, the tutorials are really, they're short. You don't see me. It's all voiceover. It's worth it. John Berger wrote, "We only see what we look at. To look is an act of choice." I think it's our obligation to get those outside of our world to start looking. Thank you.